It is October 1st. It is 2025, and this is Biology 105. Coincidence? I think not. All right. Thank you all for making it to another lecture. We're going to crush our discussion of cell membranes today. Then we're going to get into energetics and enzymes either today or tomorrow. We're almost back on track with our planned schedule, which is so cool, so great. This week in lab, you're doing some dang microscopy, you're doing some dang gram staining. It's super important. It's cool stuff. You're going to look at your cheek cells. You're going to look at bacterial cells. You're going to do gram staining and look at bacterial cells again. You're going to be characterizing some different bacteria which tend to develop antibiotic resistance. These are non-pathogenic strains that should not have antibiotic resistance, all right? But we still want to be nice and careful with them, all right? So you're going to look at these. It's going to be really useful to know how to do gram staining. You might get into a situation where you have to pay attention to the microbial community. Microbial community is usually discounted or oftentimes discounted by biologists, ecologists, but hey, guess what? We talked about endosymbiosis. There are endosymbiotes, endoparasites in most living things, and they are very rarely studied. You want to make that your field of research? You can make a whole career out of that, all right? Super interesting stuff. I'm going to stop talking about it because I want to talk about membranes. Just make sure you print your lab handout, you go to the lab. It's going to be a nice, pretty straightforward one. You get to use fire to heat fix cells. Anytime you get to take the Bunsen burners out, it's a good lab, in my opinion. So I hope you enjoy that. All right, otherwise, maintain progress on reading quizzes, maintain progress reviewing course material, and let's go ahead and get into membranes. We were talking about membrane fluidity and cholesterol as a fluidity buffer. Any last second questions before I just get us moseying? Okay, let's mosey. So last time I was talking to you about the degree of saturation of the fatty acid chains in the phospholipids, remember? And if you have unsaturated cis fatty acids, they make, I just banged the microphone, sorry, anybody listening. They make a little bend in the fatty acid chain, preventing tight packing. So more unsaturated cis fatty acids, all right, a more fluid membrane. More saturated fatty acids or transunsaturated fatty acids, but think about it more as saturated versus unsaturated, that's fine. More saturated packs more tightly, more viscous or thick or stable, okay? So you want the viscous membrane when you're experiencing hot temperatures so the thing doesn't fall apart in the heat, and you want the fluid membrane when the temperatures are colder so that the membrane doesn't freeze and crack and explode. All right? Question?
So is this, in terms of temperatures, just happening on a scale of like human body temperature? Like when it's a hot day, are my cell membranes
Great question. Great question. No. So it's not an acclimation process like that. There's a really cool article. Why isn't my clicker clicking? Did I not plug the thing in? Did I leave the clicker piece? I always forget it. Okay. It's in my other classroom. There's this really cool article here, okay? Blood lipids in Antarctic and in temperate water fish species. It talks about free floating lipids in their blood as well as cell membrane phospholipids, okay? And this is more if you grow up in an area that has very cold temperatures, you can have the more fluid saturation, but it's not an acclimation response that happens on like day to day or season to season. It's more of a,
Evolutionary?
It's evolutionary, but also it can adjust. So if a population, it's more in development. It's in developmental regulation, okay? It takes longer periods of time for the saturation levels to change. Now, it varies from species to species how much they can change this, all right? So this is an example, and I talked about this in another one of my classes, I don't think I talked about it here, of how the capacity for acclimation can be evolved, which is a weird concept of the ability you have to change your membrane fluidity is genetically ingrained. And so if it's a species that goes from a hot to a cold environment in its lifespan, it needs to have a high capacity for changing the phospholipid composition, all right? This is outside the scope of our class, something that we need to really worry about. The answer to your question is no. It is not on a hot day your body will change lipids. Cool? I'm going to try to stick more to the answers and not get too caught up in it. So here's this cool article, all right? It's all about how species of fish that live in different climates have different lipid compositions. And the takeaway of it is really that you end up with, in Antarctic fish membranes, there are high levels of unsaturated fats. All right, cool. In temperate water fish membranes, they have mainly saturated fatty acid composition. Okay, cool enough? Check this out. Temperate species have higher plasma phospholipids and cholesterol concentrations. Okay, and that kind of makes sense too, because you're in a temperate climate, right? Variability, you need to have a cholesterol buffer. That's one way of getting around that question.
Is this why fish like Alaskan salmon have very high levels of omega-3 fatty acids, while fish like tropical bluefin tuna have like the saturated fat parts that people really like to eat?
Yeah, 100%. This is a major factor in that. More unsaturated fatty acids for the colder fish. Omega-3 fatty acids are unsaturated fatty acids. Yeah, sick. Points. You get 3 John points. I'm very stoked that you put those pieces together. Great. Good. So this is just more summary stuff from that article. I used to it was terrible. Like 2 years ago, I used to teach this section and I would just put this up and be like, Antarctic fish live in very cold water. I learned my lesson. Now I have summaries for you. Okay, so good. This last slide right here is just one of those where I say, it's not as simple as just unsaturated versus saturated fatty acids. Maintaining membrane fluidity is super important, okay? Fluidity is influenced by fatty acid saturation and lipid head group composition. If you want to learn more about that, check this article out, all right? It should be open access by now. It's a 2009 article. You'll be able to find it or through the library. It's a good article. It's cool. But what I want you to worry about is just the saturation levels. Are we cool with that? Anybody got any questions about that stuff? Yeah, please. Yes, so I am not positive about, I don't know how much humans vary, but there's plenty of variability in members of the same fish species, for example, based on where they thrive, where they have been living. And this can be evolved, or it can be a process of acclimation where it changes within the same generation, but it's usually a larger time scale thing. Cool? Awesome. So yes, the answer to your question, yes. The clicker doesn't work, John. You look like a dang fool pressing this button in front of everybody. Everybody's gonna think you're lame, dude, stop. All right, so we've been talking about the proteins that get embedded in the phospholipid bilayer a little bit, right? And what's the main thing that I've been focusing on? Oh, who remembers, what's the main thing about like, what lets one of these get embedded in the phospholipid bilayer? Yeah, yes, okay, yeah, hydrophobic and hydrophilic, but let's just say regions. Awesome, points for you. So check it out, for this thing to be embedded, remember, the phospholipid bilayer, the biggest, most important thing about the phospholipid bilayer is this domain, this region of hydrophobicity. It's the one thing to know. It is probably, if you got one takeaway about cells at all, from the entire 3 weeks we talk about them, please remember that the membranes have a hydrophobic domain And just remember that. All right? And then remember, like dissolves like. Hydrophilic things won't cross. All right? Hydrophobic things will freely cross. Hydrophobic things can be embedded in the membrane. All right? It all follows if you remember that the membrane has a big hydrophobic region, okay? And that like dissolves like. Hydrophobic things go into membrane. So check this out, right? Peripheral proteins are bound to the surface. They're probably made mainly up of hydrophilic amino acids. Integral proteins penetrate to the hydrophobic core. All right? And you've all watched the film The Core, right? Where they go to the center of the Earth and they have to reset the Earth, you know, by blowing up a nuclear bomb. Just go ahead and watch it. Is DJ Qualls in that? Does anybody remember? I am 36 years old. All right, very good. So integral proteins, okay? They penetrate into the hydrophobic domain, meaning they gotta have some region of hydrophobicity. Nicely done. So they're made up of hydrophobic amino acids. For some integral proteins are transmembrane proteins. So not only do they penetrate into the hydrophobic core, like right here, but they span the entire membrane. And that's important for if you're trying to transport something or if you're trying to read a signal outside and transmit it to the inside. You want a protein that spans the whole region. So, for a transmembrane protein to remain embedded in the phospholipid bilayer, it has to have a hydrophobic region here and then two hydrophilic regions on either end. Remember how I talked about that on Monday too? And I seem really, really obsessed with this. Do you think it might be on your next exam? Probably. Yeah, probably. Why not? Right? Okay. Whenever I get super obsessed about something like that, it's usually because it's a question people always miss on exams. And I want you all to get that question right, because it means people don't understand it. And it's a really important concept. Anyways, membrane proteins are important. Sometimes we represent the importance of a given type of protein based on how much of the genome is dedicated to encoding it. So if 25% of genes, look at humans, almost 30% of your genes exist just to encode membrane proteins. So membrane proteins, what does that tell us? They're important, and there's probably a lot of variability and a lot of different functions they have. So let's talk about some. Why not? The clicker. All right, here are some different things that membrane proteins do. We are going to talk about cell-to-cell recognition. We will talk about signal transduction quite a bit, and we will talk about transport quite a bit. So A, C, and D on this figure, we're going to be talking about. They're good things to be focusing on. Intercellular joining, we've already talked about it to the extent that I'm going to discuss it. Unless of course, you have any questions. But it basically sticks cells together because you want the cells to remain where they are. Attachment to the cytoskeleton and the extracellular matrix, same thing. We've talked about it. It stabilizes the cells where they are. That's all we're going to worry about. Them as enzymes. Question, please. Oh, sorry. It's A, C, and D. Those are the ones that we're going to be talking about in a lot more detail. Yes. And really, A and C are the ones that we're going to talk about the most. Cool. So these two, we've already talked about them when we discussed the extracellular matrix. We're good, okay? Membrane proteins as enzymes. We're going to talk about enzymes a lot, but all this is here to say is that, hey, sometimes enzymes are also stuck in the membrane. Okay? We're going to talk ad nauseum about enzymes. You're going to get sick and tired of talking about enzymes. Sometimes an enzyme is in a phospholipid bilayer. Okay? And there's some really cool cell-cell signaling stuff where, like, sometimes one of these enzymes will actually break down a phospholipid to serve as a signaling molecule after it recognizes something outside. So it's like an overlap of a It's got overlap between this and this, but, or sorry, this and this. But fine, we'll talk about it later. We'll talk about it later, okay? Transport. If something needs to be moved across the phospholipid bilayer, you use a dang protein. If it won't usually freely cross the phospholipid bilayer. And we're going to talk about which types of molecules freely cross the phospholipid bilayer and which types of molecules do not freely cross the phospholipid bilayer. And it is all based around the hydrophobic domain of the phospholipid bilayer. So important. All right. Some figures, okay? Cell-to-cell recognition. This stuff matters. Here's an example of how HIV hijacks your cell's receptors and tricks the cells into thinking, we want that. That is something that we want, okay? So you've got these two receptors, okay? They're two different proteins that sort of work together to receive a signal. They're both encoded by a separate gene. They're separate proteins, right? Very cool. When you have both of those genes, you have a functioning receptor. That receptor can identify a carbohydrate label coming off of a glycoprotein embedded in a vesicle. So here's a vesicle with a phospholipid bilayer that is projecting, okay, these carbohydrate labels. If the receptor receives that carbohydrate label, it says, this is from me, this is good, we need to bring this into the cell, okay? Because otherwise, the virus can't enter into the cell. So it's got this little case. If the receptors register with that carbohydrate label, which is really a glycoprotein with the carbohydrate portion, then they merge, they fuse that membrane with the other membrane and bring the virus in, okay? If you're missing one or both of those receptor genes, okay, or missing one or both of those receptors, then the HIV can't be detected, its little sheath, and so the cell doesn't detect it as wanting it, doesn't bring it in. All right? So this, if there are people who do not have this gene, okay, for the co-receptor, and therefore they have a natural resistance to HIV. Kind of cool. Question?
So are the glycoproteins making the receptors?
These are two, I might have misheard you. These are two different proteins making up the receptors. Was that your question?
So each protein is an individual receptor?
Oh, solid. So we've got a receptor and a co-receptor, which means that they only are an effective receptor when they occur together. You need both of them. So each one is sort of making its own receptor, but they can't work on their own. So I wouldn't say it's super accurate to be like, oh, even though this is called a receptor, that's just terminology, right? So in the common vernacular, it wouldn't be as accurate to say, oh, they're both receptors because they can't receive the signal without the other one. It's actually a receptor-co-receptor situation where in order to function, you need the receptor and the co-receptor. Did that make sense, or did I muddy the waters more? Oh, please, please, please.
That was important to learn too. But
Okay, cool.
I guess my question is, so there's several receptors. Are each of them an individual protein, or is the protein containing several receptors on it?
Got it. So in this example right here, we have a single protein as a receptor, all right? In this example here, we have two separate proteins as a receptor. So yes and no, right? It can happen either way, okay? And then these proteins also can have primary, secondary, tertiary, and quaternary structure. So each one of these, this co-receptor could be made up of multiple polypeptide chains that had to come together to make that receptor. So the unsatisfying answer is it's complicated, you know? Yeah. Awesome. Please, question.
I'm a little confused. But to summarize, one of the proteins, or multiple of the proteins, have receptor, co-receptor, and another protein or thing binds to it and then fuses with the cell.
Here's what it is, right?
What's the summary, I guess?
The summary is there are receptor proteins that can detect signals from outside, right? Like this, okay? And there are other proteins that project signals. They're usually glycoproteins, and they stick out some sort of carbohydrate label, and that gets detected by the other receptor. So this receptor allows the cell to say, oh, that's a friendly cell, or that's not a friendly cell. So this example specifically of cell-to-cell recognition is really just another sort of signal question.
They signal to each other and then fuse,
Basically.
So kind of like neurons, I guess?
Kind of. Not, yeah, not, let's just go with kind of.
Okay.
Solid.
That makes more sense. Thank you.
Awesome. Good to hear. Yeah. So here's the thing. It's like here's the real takeaway, right? If you need to detect a signal outside of the cell, you do it with a membrane protein. The membrane itself can't detect a signal really, right? It's just a bunch of phospholipids. But having a special protein, you can detect a signal and trigger a response in the cell. Okay, cool. Very good. Oh, and if you don't have the receptor, you can't receive the signal. That's the last piece from this example here. Is that okay? Anybody got any additional questions? No need to worry if you do. Cool. I'm not even going to bother making you answer this because I have talked about it so much. All right? Say. Right. Good. Okay, cool. All right. Membranes are something that we call selectively permeable. Certain things can cross, certain things can't cross. Usually, you'll be taught a semi-permeable membrane or a selectively permeable membrane as it only lets water cross. Has anybody encountered it in that way? Of like, oh, look at the semi-permeable membrane for osmosis and water crosses, but nothing else can cross. Yeah? We've heard it that way. Inaccurate, not true. Other things can cross selectively permeable membranes, okay? It's all about the hydrophobic domain. Are you sick of me saying hydrophobic domain yet? Because I've said it a bunch. But this is important because it determines what can cross, okay? And if the thing can't cross, but the cell still wants to move it across, it's gonna need a membrane protein. Or it's going to need to carry out exocytosis or endocytosis, where you make a little vesicle and bleb it off of the membrane and ship it off. Exactly, it's exactly the move. It looks like this, and the cell does it. All right, so let's talk about relative permeabilities. I love this figure. I love it so much. I love when a figure summarizes things so effectively. And if I love a figure this much, I'm going to give it the due respect of putting a question on the exam associated with said figure. So, big important figure. All right? Look at it. It's beautiful. Check it out. Small hydrophobic molecules and gases, or molecules made up of nonpolar covalent bonds, they're hydrophobic, right? They're going to diffuse really rapidly across this. They're small molecules, and they're hydrophobic, so they're just totally soluble with all these fatty acids. Reasonable enough? So oxygen, carbon dioxide, nitrogen gas, little tiny hydrocarbons, those are all small nonpolar molecules, and they should diffuse really rapidly. Cool? We've already spoken in your lab about some rules of diffusion, right? How things move from areas of high concentration to areas of low concentration. How does everybody feel? Or are there any questions about diffusion in general? All right, cool. It's one of my favorite things, so I miss being in the lab and getting to talk to you all about osmosis and diffusion more. Hopefully you've been receiving enough information, but you can always come to the office hours and talk to me about them if you have any questions. Anyways, moving forward. There are some small polar molecules that can cross the phospholipid bilayer. Now remember, this is all diffusion, so it's all dependent on relative concentrations, okay? Water can cross the phospholipid bilayer, and it does so at a decent rate, right? Not as fast as oxygen will diffuse across, right? But it's because it's so small and it's everywhere. So even though water is partially repelled by this hydrophobic domain right here, it will still cross. There's just too much water, and everything's moving around. And sometimes water's going to have enough energy and just go, brr, boom. All you got to do really is make it halfway through, right? You make it halfway through, then you're just as likely to be rejected out this way as this way. See what I mean? Yeah. That's just that same way with life, right? You just got to get, I don't know. Ridiculous. Large, uncharged polar molecules will be rejected, all right? They're too big, and they're polar molecules. They're not going to pass across the phospholipid bilayer, except for rarely. So glucose, sucrose, they got a lot of polarity. They have a degree of polarity in the molecules, all right? And they're also pretty big molecules. They'll still cross every once in a while. This is all, you know, it's just a thin sheet of oil. It's not going to be perfect at repelling everything, okay? But it's good enough to set up order. Now, here's the thing. If this was not true, we could not survive. If this becomes not true because of a poison that gets into your body, you perish, all right? This is hugely important. Ions, they will not cross a phospholipid bilayer at any appreciable rate, regardless of the concentration gradient. You can have a huge concentration gradient of ions across a phospholipid bilayer, and they will be aggressively repelled. Think about a charged molecule, an ion, as being super hydrophilic, because the water molecules are just going to surround it, you know? Like if there's a positively charged molecule, okay, even if it's small, even if it's just hydrogen, water is still going to aggressively surround it, right? Because of how polar water is. So ions are essentially like super hydrophilic. And is that an oversimplification? Maybe, but it works pretty darn well. Cool? That makes sense to folks? At the back, question.
You mentioned that like a poison could make it so they cross.
Yes.
Like what specific poison?
Oh my gosh, what's the name of the big one? What's the name of the big one? It's going to come up in our cellular respiration one, but there are a number of both pesticides, herbicides, and chemical warfare poisons that only work by making membranes permeable to ions. And I'm not remembering the names right now. I'm so sorry.
Cyanide one by chance?
I don't recall. I mean, I want to say like I think so, but I'm not confident enough in my answer to say it on record, you know. It's a good guess. Yeah. Yeah. That's, you know what? We're all here. We're all having a good time. We're all enjoying Biology 105. Let's look for poisons which work by increasing membrane permeability to ions. I want something fun. These aren't fun. This is like a research article. Just give me an image. That'll usually work. It'll be like a bunch of pictures. This is boring. I blew it. I messed it up. Sorry, everybody. Examples of. Are you going to trust this AI overview? I don't think I am going to trust it. Whatever. I failed. There's a lot of really cool examples of him. Yeah. I forget if like Agent Orange was one of them. It might be. Anyways, pretend that didn't happen. All right? I was assuming it'd be way faster to find some good sources. But I do have a slide coming up. Yes? DDT. Thank you. Thank you so much. John points. 4 John points for you. DDT. That's a big famous one, right? Absolutely. Ah, that's great. Okay, so if the molecule can't cross by diffusion, or if you want the molecule to cross more rapidly, or if you want the molecule to move against concentration gradients, you're gonna need a membrane protein. All right. How good is this marker? Yeah, it's not good enough. I'm not going to do it. All right. So check this out, right? We've got the process of passive transport. We've got the process of active transport. Passive transport is diffusion, okay? Passive transport relies on concentration gradients. I'm going to do this. I'm going to do this because this is one of those things that's super important, and I think that I tend to rely too much on just saying it. It'd be okay to write some stuff down, but it's too small on this. Anyways, let's zoom it in. How'd I do? My nails are pretty good today. I'm not feeling too, that's not like too gross, right? Looks pretty good, pretty good. Okay. So we've got passive transport. Is that legible at all? Pretty good. Here we go. No energy required. All right, no energy required for passive transport. Boom, boom, boom. Okay? Things move from areas of high concentration to areas of low concentration. These square brackets right here indicate concentration. Example, concentration of sodium chloride. You will see these square brackets used all the time. It's good to get used to using them in your own notes. You know, it's faster to write, okay? So passive transport, no energy required, all right? Things we move to high to low concentration, that's awesome. And it may be facilitated by a channel, all right? So basically, that's just like a protein that's open, like this, that lets things diffuse through it. It makes it more rapid than just relying on it diffusing across the phospholipid bilayer. So there's an example of that, of aquaporins, which are just channels that specifically let water cross membranes more rapidly. 1 second here, I'll fix that. So there's passive transport. How's that? Question?
So the channel that you're talking about, is that be considered a microtubule or no?
No, but solid. It is a microtube, but it's not made of microtubules. Though, to be fair, my like protein structure knowledge is limited to like pretty basic understandings. So this should not have any microtubules in it, but microtubules are made of proteins. There may be a structure in here that's similar to a microtubule, but I don't think it's going to be tubulin, so it won't technically be a microtubule. Yeah. Yeah, I did a pretty good job of hedging my bets there, huh? Solid. So check it out, right? Diffusion, the thing just diffuses across the membrane. In facilitated diffusion, there is some membrane protein facilitating the diffusion, but it's still following rules of diffusion, where molecules are moving from areas of high concentration to areas of low concentration. So the key here is there's no energy required, that's what makes it passive, and the direction of the movement. Those are those key points. I put stars next to them. Question? The channels can be opened and closed. So there are these things called ligand-gated ion channels, for example, which it basically just means it's a channel that has a gate on it. It's gated. A ligand is something that binds to another thing. So when something binds to the gated ion channel, it opens, and it lets ions just flow through it, right? So that is an example of a channel protein that has a gate on it to open or close. And this here, some of these proteins bind to the molecule temporarily, and that's like how they facilitate the movement across. For our purposes, let's focus mainly on this example of facilitated diffusion, if that's cool with everybody. Yes, please. Oh, sorry, real quick. Did I answer your question? Yes. Awesome. Now, yes, please.
Sorry.
No need.
So this flow of no energy required is from concentration gradients. What about electrical gradients, or is it just concentration gradients?
So think about it. If these are charged particles, then it's an electrical gradient.
Okay, so
Yep.
charged will be electric gradient, non-charged concentration.
Yep, and it's still a concentration gradient. So
It's
electrical. Yep. So the concentration gradient is like the hedge your bets more general terminology because that includes if they're charged particles. They're still reliant on a concentration gradient, and that's just generating a voltage. Yeah, solid. I love it. Additional questions about this? We're going to talk about active transport now, and I'll just do this. Active transport. I love this document camera. It's really cool. All right. Energy required. All right, I'm going to write yes, energy required, okay, to contrast with no energy required. Molecules move from areas of areas of high concentration, areas of low concentration to areas of high concentration. All right, so that's why energy is required, okay? Every system wants to fall to equilibrium. Big quotation marks around wants. Concentrations want to be the same across membranes. Okay. All right, cool so far? All right, now of course the system doesn't want anything. This is a natural outcome of physics. But, so I want to also write out a couple of other important things here. This right here is against a concentration, I'm going to write the word concentration this time, against a concentration gradient. Whereas this right here, that is down a concentration gradient. That's just a terminology thing. So in passive transport, molecules move from areas of high to areas of low concentration, or particles, let's say, because they're not always full molecules, they can be ions. This is down a concentration gradient. It requires no energy. It might be facilitated by a channel protein, all right, or a membrane protein. In active transport, energy is required because you are moving against a concentration gradient. Sometimes people will say up a concentration gradient, but against, I think, is better language because it indicates that you're moving against the natural tendency, which is for the system to fall to equilibrium and for concentrations to be equal across the membrane. Question?
Looking at the diagram, how it has like the two different sides, I didn't know if you were going to go over the diagram specifically.
Yeah.
Is that showing that it will like pick and choose which it lets through? Because also the two different arrows are different colors for the square particles that it's letting through and the circular particles that it's...
Totally. Yeah, totally. So this is like an example of a sodium-potassium pump. And the sodium-potassium pump, if you're doing a physiology course, is really, really, really important, okay? Like you'll talk about it in ZO 110 if you take that course, okay? So that's why it's their example of active transport in the textbook, and this is taken from, the textbook. But check it out. So yeah, what's happening here? The orange arrow is indicating that the orange stuff is being pumped in this direction against the concentration gradient from an area of low to high concentration. It's active transport. This transporter also pumps yellow stuff, all right? Consuming energy in the same way, same process. It's more efficient, right? Because you want to be pumping both of those to set up a concentration gradient of both. But it has to do with a lot of like neural signaling and muscles triggering. And so we're just not going to, worry about it too much here. But did I answer your question okay? Yeah. Solid. So it's just pumping in both directions, but it's always pumping against the concentration gradient, and it's consuming energy in the form of ATP. Question.
So I guess you actually just answered it. Last few words.
I'm so good.
When you say energy required, that energy is ATP.
The energy is provided by ATP in almost every case. Yeah, no problem. I love it. Additional question. How is the ATP consumed for the process? Yeah. Okay, yeah. So ATP in the abstract is pretty mellow, but the more involved you get with talking about ATP, the more complicated it gets. So targeting anything inside of a cell is outside of my expertise, right? Like how does it know to put it there? It's like ATP's in high abundance throughout the cell is one thing, right? And then there are enzymes that are involved in breaking this ATP down and transferring the energy to this. And when you transfer the energy, it usually comes from taking that terminal phosphate group. Like here, I'm ATP right now, and this is my terminal phosphate group. And then there's an enzyme that goes, okay, take that off and put it on this thing. And that makes this thing change shape, all right? And now it's a different shape. So now it'll do something different, like move those things around, all right? And so that's how I just pumped something. I turned it into a proton pump. You see, did that work at all? Kind of? Okay, sick. So usually, this is all accomplished by, is the enzyme there at the right time, okay? And is the ATP present to carry out the process? And then there's regulation of the enzyme, which can be direct through concentration of these solutes, right? So like imagine that there's a peripheral protein in association with this transport protein, and that peripheral protein is an enzyme that is regulated by the concentrations of the orange stuff. And when the orange stuff levels are low, it hydrolyzes ATP, attaches the phosphate group to this thing, and makes it change its shape. And when it changes its shape, it's pumping things. How's that? Cool. So the unsatisfying answer, as always, is very complicated. Yes?
It can change its shape based on a peripheral enzyme to pump different stuff.
Yeah. It's kind of wild because it's like this, right? When it's open to one side, it exposes these sites that can grab something. So now it grabs the thing, and now that thing is bound. And then it's just going to open when it changes form again towards the inside, and then it drops it inside. When it changes its shape, it both changes which side is open, and it changes these sites so they no longer want orange. Now they want yellow. So when it's open to the inside, it wants yellow, and when it's open to the outside, it wants orange. So that means that every time you do this, you're going to take orange and move it in, and yellow and move it out. Yeah, because the shape change is, it's all accomplished by shape change. Awesome. And that's one of the ways that these proteins wear out over time, right? Is they have to change shape all the time. That ended up being a dang fruitful discussion. I liked that. Thank you for your participation. Both of you can have some John points. I'll give you 3 because you got 4 and I really liked yours. I'm so sorry. Okay. We're going to just go ahead and crush this transport protein stuff. I'll leave this here, and if we want to take a picture of it, I guess you could. I don't know. Let's go ahead and do this. We got a few more minutes, right? Like 2 more minutes? I'll use 2 more minutes. Okay. Transport proteins. What do? They transport things across the membrane. So they got to be transmembrane proteins, right? They have to span the entire membrane to transport things across the membrane. Solid? Okay, some are aquaporins. These facilitate the passage of water molecules. Channel proteins, okay, check it out. If this channel is hydrophilic, well, then hydrophilic molecules are going to be able to cross because they sort of dissolve into the hydrophilic region. Here's a little video, if it works, of an aquaporin. These are water molecules. And see, the amino acids that are in the center of the channel, they have some affinity for water. They're hydrophilic. So the water molecules aren't repelled. So the water molecules can cross way more rapidly. It's still along concentration gradients. It's still passive. And look, every once in a while, a water molecule tries to jump into the phospholipid bilayer, and it just goes like, no, and it gets repelled. This one's trying to make it. It's trying to make it, but not quite. All right? So that's an aquaporin in action. Any questions about channel proteins? Yes, please.
So noticing the example, it seems like the water is trying to go in through both directions.
Yes.
I thought they were generally trying to flow from like one concentration to another.
I'm going to give you so many freaking John points. This is great. Here's the thing about all of this diffusion stuff. All of these molecules are moving in random directions. There's no directionality to it. But when there's a concentration gradient across a membrane, just the sheer number of molecules here means that the likelihood of something going in this direction is higher than the likelihood of something going in this direction, even though for each molecule, the likelihood of it moving in any direction is equivalent.
It's not necessarily being pulled into a low concentration. It's more like simply the odds are higher.
100%, 5 John points for you. It's not that things are being pulled towards a low concentration area or pumped. It's that just by chance, the odds are things are going to move towards lower concentration areas. And that's where we'll come back to on Friday with our very exciting discussion of osmosis. I know you already got it once in lab. I'll do it again here because I can't help myself. Have a wonderful day, everybody. I'll see you on Friday. Yeah, farewell, farewell. Yeah.
So is water displaced?